Let's meet the algae. Do you know what algae are? First, I'll explain about algae. Algae are autotrophic and they undergo photosynthesis to make food. They are eukaryotic. Microalgae is an algae which is single-celled, and giant kelp is formed by multi-celled algae. Algae are aquatic organisms, so they have to live in water. By the way, did you know that algae are used in many products? They are used in cosmetics, toothpaste, fertilizers, chemical, and food, like sweets, and even in chocolate milk. What's more, they are a sustainable source of energy. Algae are very useful for mankind.
Now I'll tell you about how we found algae and show some pictures of them. To begin with, we walked around Cambridge. This map shows where the algae are. This is pond, this is on the tree, and this is near the Cam River. And then when we back to the laboratory, we filtered the samples and used light microscopes to find algae from water. So now I'll show you some pictures of them. This is green algae, which is simple single-celled. This is Pediastrum. This is Scenedesmus. And this is Oscillatoria. It's a brown algae. And this is cyanobacteria, but this is not algae.
So how is algae formed? Algae is formed by a process called endosymbiotic. What it is the cyanobacteria, which we've just been shown previously, is engulfed by a eukaryote. The cyanobacteria is a prokaryote. It's not digested, but it remains in the eukaryote. So later, with evolution, this led to green algae and red algae. That was primary endosymbiosis, but we also have secondary endosymbiosis. What this is the product from the primary endosymbiosis was engulfed again by another eukaryote, and this led to further developments and also brown algae and diatoms, which are a type of brown algae. How we showed these different pigments was we done a chromatography sample. On the left you can see is the brown algae, and on the right is the green algae. We done this by placing a dot of concentrated algae, which we done in a centrifuge, on the baseline and place this in a solvent. As you can see, there are two marks at the top, and this shows that they're the same pigment. So brown and green algae both have a similar pigment, but brown algae, which evolved later, has this different pigment which green algae don't have, which shows its evolution. We also done an absorption spectrum to identify these pigments.
These algae is chlamydomonas. If we look at them through the light microscope, we can see a circular structure. This circular structure is perinoid. This is a TEM of the algae. There are the top of the picture is the nucleus, and perinoid plays a very important role in the photosynthesis of algae. Then I'll talk about perinoid. Every plant, such as algae, consumes CO2 for photosynthesis. But in the water, CO2 reacts with H2O, like this. And most of them exist as HCO3-. Algae can't use HCO3- for photosynthesis. Then algae transport the HCO3 for actively through the transporters in their membrane. And then carbonic anhydrate occur, and HCO3 is changed into CO2. But CO2 is up to spread. Then the pyranid work effectively. CO2 is transported to pyranid. Pyranid work as a box, and it stores CO2. So the algae can use CO2 for photosynthesis and can make sugars.
Please look at these diagrams. They are simple models of algae. The black dots represent Rubisco, which is an enzyme for carbon dioxide fixation into sugar. And this yellow box is pyranid. Which is more effective, 1 or 2? Number 1. Number 1 is a wild type, and it has a pyranid with highly concentrated Rubisco. In water, carbon dioxide diffuses easier, and pyranid helps to accumulate carbon dioxide made from bicarbonate. That's why it's more efficient. Number 2 is a higher plant, and it has no pyranid. It's less concentrated. Diffusion lasts longer. Carbon dioxide can escape by diffusion. Carbon dioxide concentrating mechanism contains 3 components: transport of bicarbonate, carbonic anhydrase, which makes carbon dioxide from bicarbonate, and pyranid, which is a box to accumulate carbon dioxide. Pyranid is a booster for photosynthesis.
Okay, so we're going to have a look at the Rubisco enzyme structure, and that's the enzyme involved in carbon fixation. As we can probably see a bit, 8 large subunits and 8 smaller subunits, and on the smaller subunits we have alpha helices, and those are important because if we replace the alpha helices of algae Rubisco with that of higher plants, so if we take away the green parts, and put the alpha helices from a higher plant, so for example spinach, if we change them, but the structure, the Rubisco enzyme is still the one found in algae, that would mean that it does not have a pyranid, so then it lacks the pyranid structure, which is basically a bad thing for the plant. Okay, so photosynthesis. Under natural light conditions, chlorophyll molecules absorb photons, becoming chemically excited and unstable, so they must dissipate the energy either through photosynthesis or as heat, or the other part is fluorescence, so that's when it re-emits photons of a lower frequency and wavelength. So the experiment that we did, we wanted to investigate the efficiency of photosynthesis using a PAM fluorescence analyzer. So in both wild-type green algae and mutants which had their helices replaced with those of higher plants, so for example spinach. So samples were placed in labeled cuvettes and kept in dark conditions. So the PAM fluorescence analyzer assesses photosynthesis efficiency. So first, the PAM produces a measuring light, which is not intense enough to stimulate photosynthesis, but it does promote a fluorescent signal, so that would be termed FO, as we can see, which represents the open photosynthesis reaction centers. So that has the ability to photosynthesize, but the light's not enough. And the second beam of light that's transmitted is very intense, and it closes these photosynthesis reaction centers, so fluorescence becomes the maximum because photosynthesis cannot take place. Because energy can't be used up in photosynthesis, so it needs to be dissipated in another way, in another pathway. So using the equation shown, this produces a ratio which is a relative measure of the number of photosynthesis reaction centers available. So we term that the quantum yield efficiency of photosynthesis. Okay. So if you look at the results, this is our data, and the higher the ratio, so the higher the bar is, the better the plant is at photosynthesizing. So the wild type algae represented the highest ratio, so that's the WT, and this had the pyronoid structure inside it. But you did not possess the pyronoid, so they were less photosynthetically efficient. So in conclusion, we wild type and mutant algae possess the same amount of chlorophyll and rubisco, and this has been experimentally determined previously. So therefore it can only be the pyronoid which affects photosynthetic efficiency. And this is what we're trying to investigate, and they have an ongoing project in Cambridge about this, looking into pyronoid structures. So we'll look at applications now for the future. So Chlamydomonas, that's the green algae that we used. We used that particular type because all its genome sequence is known. So we can easily incorporate genes into higher terrestrial plants to make these, so the alpha helices in algae, we can take those and put them in higher plants to make something like the pyronoid. So that would mean that the plants who would have this structure in their rubisco are more photosynthetically efficient. Therefore, they can grow faster because they can concentrate their carbon more and fix sugars at a faster rate. So that would be very beneficial for us because we can have food for the growing population. And also decreases carbon dioxide levels in the atmosphere, and therefore reduces the greenhouse effect. And it benefits a lot of people, so we thought we'd just sum it up.
Lastly, special thanks to Moritz Mayer, Jessica Finch, Richard Perez Story, Danny Wood, Elena, and Callum for all of your help and assistance given. Thank you for listening.